I would like to thank the organizers for the opportunity to share information on the licensure of veterinary biologics with you today. For this portion of the meeting, we'll cover the vaccine approval process as it relates to licensure of salmonella vaccines by poultry. Next slide. I work for the Center for Veterinary Biologics, which is located in Ames, Iowa. We're the regulatory group in the USDA that does the licensing, testing, release, and compliance work for veterinary biologics. CVB is divided into three functions: inspection and compliance, which inspects the companies that make veterinary biologics. This group releases the serials of product to the market. They are also the compliance group for the program. They work with the products predominantly after licensure. The laboratory that tests veterinary biologics both before and after licensure. I am part of the review staff that evaluates data and information supplied by the firms to determine if a license may be issued. Most of our work is done prior to licensure, but we work with the firms and the products post-licensure if there are changes for the product. One type of product that we regulate would be salmonella vaccines for poultry. Next slide. On this slide, you will see the codified definition of a veterinary biologics. You can see it's quite long and complicated. When I started with CVB, we did not consider this definition to include pre-harvest food safety vaccines. However, in 2005, we interpreted it to include managing a carrier state for some pathogens, which led an opening for this type of product. Next slide. The basic licensure steps begin with the purity and identity test of a seed. The firm conducts this testing, which is confirmed by our laboratory. The next step would be to develop a vaccine production process or recipe to make the product. Then the firm would show efficacy of the product in what is usually a vaccination challenge experiment. During the licensure process, they demonstrate safety of the vaccine. Next, the potency test is correlated to efficacy and validated. During the licensure process, after the validation of the assay, each serial or lot of vaccine would be tested for potency. The last steps would be the production of 3 commercial-sized serials of product that are confirmatory tested by our laboratory. At least 2 of the serials would then be tested under field condition to assess safety of the product. Labeling is proved at licensure, and safety is monitored further post-licensure. Next slide. Now let's look at salmonella vaccines for poultry specifically. They may be live attenuated vaccines or killed inactivated bacterins. While CVB is generally looking at preventing or reducing disease for products, in the case of salmonella products, we're looking at a reduction in the colonization. In this case, we're licensing biologics as a pre-harvest food safety intervention. Efficacy is the reduction or elimination of the carrier state. We do not allow food safety or human health claims to be made for products under CVB authority. These claims would be under the FDA. Next slide. Let's look at killed vaccines first. For a bacterin, a master seed would be designated. This is a specific lot of bacteria grown and frozen back. It is tested for purity and identity by the firm. The firm's results are then confirmed by the CVB laboratory. The firm would then select an inactivation procedure and a test and an adjuvant to be used with the killed product. The efficacy serial is then prepared. The efficacy study is a vaccination challenge study. Various organs are evaluated for the presence of the challenge organism, with organs being scored as positive or negative. We found colony count data to be quite variable, which has led us to the positive and negative interpretation. A potency test is then devised and correlated to efficacy. This is often an ELISA-based test. The inactivation process is evaluated. and a test for each bulk is then devised. 3 pre-licensing cereals are made and tested by the firm. Samples of these cereals are then submitted to the CVB laboratory for confirmatory testing. At least 2 of the 3 cereals are then tested under field conditions for safety prior to licensure. This is generally conducted at 3 geographic locations. Next slide. Another type of Salmonella bacterin could be prepared as an autogenous product under a USDA license. Autogenous products are killed products that are returned to the flock of origin of the isolate. There's no efficacy study, no inactivation study, no potency test, no field safety study to support this type of product at the beginning. While they're only returned to the flock of origin, in general, with special authorization, they can be supplied to other flocks. The isolates are good for 24 months with request. To continue use of the isolate after 24 months, an antigenicity test is conducted and a potency test is added. And we're currently looking at changes to these policies. Next slide. For a live product, additional licensure studies are required beyond those for a killed product. We again have a master seed that is designated, but then we require some additional studies that assess the behavior of the vaccine organism in the bird. One of these is a reversion to virulence study. In this, we pass the salmonella seed through birds 5 times. At the end of these 5 passages, we look at the genotype and phenotype of the resulting organism. An overdose study, you would give the salmonella seed to the birds at a 10x, and then look for clinical signs or adverse events. A tissue tropism study, where we evaluate where the salmonella seed goes in the bird and how long it stays there. A clearance study would then look at when does the bird clear the salmonella seed or vaccine. A shed spread study, we look at does the salmonella seed or the vaccine shed to other birds. An efficacy study would then be conducted, and this would be similar to the format of the efficacy study for a killed product. And then a risk assessment with Federal Register publication is created to obtain any additional thoughts on the licensure of the vaccine. If this is published without significant comments, the licensure process moves forward to the field safety study. In the case of live salmonella products for poultry, the trial may be coordinated with FSIS. Next slide. So let's look at some more of the details for this efficacy study that's shared between the live and killed products. The efficacy study design would apply to both, and approximately 20 birds would be vaccinated. The use of younger birds at this vaccination step would imply the use of the product in broilers, while the use of older birds might imply the use in layer birds. In addition to this group, there are usually 2 control groups. One receives a placebo, and the other group receives nothing. The vaccinates and the placebo-vaccinated groups are then challenged. The uninoculated group then remains unchallenged to look at any pathogens or other things that could have been brought in with the group of birds. The type of salmonella used for the challenge determine which salmonella the label claim will supply. Each salmonella claim is supported by a vaccinate challenge study. After an observation period, the birds are euthanized. Organ samples are collected and evaluated for the presence of the challenge strain by culture. Note above that for a product given to older birds, which would be designed for layer birds, that the reproductive tract is included in the product design. Next slide. Let's look further about the risk evaluation that I mentioned. CVB originally developed the risk analysis for biotech products to assess the risks of the vaccine microorganisms to animals, public health, and the environment. We've expanded the evaluation to include impacts of the vaccine on other regulatory programs. While this was informally evaluated previously, The incorporation of a formal risk evaluation has allowed a more structured approach to this evaluation. For the live salmonella products for use in poultry, we discuss the product with the firm and other potentially impacted programs. This occurs before we do confirmatory testing of the master seed so that the firm is apprised of the concerns. The risk evaluation is published in the Federal Register prior to the field safety trial to again allow further comment on the proposed vaccine. Since most groups now employ the use of sequence data, CVB facilitates the transfer of the sequence data to the other programs when licensure is imminent. Next slide. Another type of product that you might see for a salmonella vaccine is called a platform product. It is a non-replicating, non-viable biological veterinary vaccine. It's essentially a backbone into which genes can be inserted. For the first construct, we go through the licensure process as I've described it. The licensure is then expedited for subsequent constructs. Next slide. So we've discussed salmonella vaccine approval and vaccines that include the designation of the master seed and production method. For live products, studies are conducted to evaluate the vaccine organism in the bird. Efficacy studies using a vaccinate challenge design are required with scoring of the organs as positive or negative. 3 serials are made at the production scale. The pre-licensing serials are tested at the firm, confirmed by the CVB laboratory, and used in the field under normal poultry conditions. For live products, the basic licensure process for killed is used. The licensure information is augmented by studies to understand the behavior of the vaccine organism in the bird. During the licensure process for a live salmonella product, CVB works with other regulatory programs potentially impacted with the use of the vaccine in the field. And the question then that we were supposed to evaluate is what one strategy will have an impact, make a difference in reducing salmonellosis. And to this, we would say that vaccines are one tool that can be combined with other mitigation strategies to reduce salmonellosis in poultry. Next slide. So if you have any questions, please feel free to ask them. Thank you for your attention.